The first step of a genome project was reading the DNA sequence, right? That's why DNA sequencing is the basis of understanding the structure of DNAs. So we are going to study the reading the mechanism or the reading method of DNA first. And then also another important thing is amplifying the DNA. So we are going to study the basic principle of PCR in this chapter. DNA sequencing technique has been developed different formats. One possibility is utilizing radioactive element and run 4 different gels targeting the ATCG as a separate signal. But currently most optimized technique is utilizing fluorescence molecules and the Sanger method. Sanger sequencing method has been developed in the 20th century by Frederick Sanger. And Sanger sequence has been used as a routine and standard method for DNA sequencing applications. And even though we have a new technique, so-called next generation sequencing, which has much faster the sequencing capability, still to validate the data quality, Sanger method has been used as validation method. In Sanger method, we utilize the DNA polymerase. And let's say this single-stranded DNA is the template we want to read, and we add a primer first. This primer is a short DNA sequence which can define the initiating point of DNA synthesis. And here, if you add building blocks of a new DNA synthesis, these are triphosphate of a nucleotide we have seen before, right? During the DNA synthesis, we add dideoxynucleotide. And this dideoxynucleotide can be incorporated into synthesis and it can terminate the synthesis. This unique building blocks has 2 features. One is it doesn't have 3' hydroxyl other groups. That's why it terminates the synthesis. And second of all, each of them has unique colors of fluorescent dye, and it can designate which the base is introduced. So the synthetic product of this synthesis is 4 different color labeled different size of DNA fragment. The mixture of these the DNA fragments altogether, it can be injected into capillary electrophoresis, and depending on those size, they can be separated. At the end of this capillary electrophoresis, there is a fluorescence detector, and it can start to read those fluorescence from those fragments. By reading this sequence, now it is possible to read the whole sequence of this DNA. The first stage of a human genome project has been done basically by this Sanger method. As you have seen, Sanger method is quite automated system. Still, you need to manage each sample one by one. Now, the sequencing technology has been developed further and further. Now we have a totally new concept of real high-throughput DNA sequencing method we could get. This new method we call NGS. NGS sounds like familiar to you, right? It's not NUS graduate scholarship. That's a next generation sequencing. Now let's look at the mechanism of NGS. In NGS systems, now all the DNA samples are in parallel prepared, utilizing some specific enzyme cleaved off, and both sides is labeled with specific sequences which can match to the primer, and these 2 primers can be loaded onto the chip. So a lot of the 2 primer is loaded onto this chip. And then mixtures of this the DNA sample is added. Then at least one side can be bound to one of those the primer, and then DNA synthesis can happen. Now denaturate, then the original sample is removed, and the next cycle it goes into bind to the other side of the primer, and then DNA synthesis can happen following this way. Then it finish up those syntheses here. Now if you denature it, now both of the strand is bound to the surface here, and one was original sequence and the other is a complementary sequence, and this process can be repeated because there are a lot of the primers waiting on the bottom. That's why each unit of this. This is same sequence of the same sequence of original the DNA samples. That's why this kind of the identical sequence sample we call clone. Then each cluster here, those are representing different DNA sequences. Then next job is to read out those sequence of this DNA for each individual the cluster. After denature of this the double helical strand and then cleave off one of them, and then one sequence is removed and only one forward sequence remained here. Now here the sequencing started. Utilizing the primary sequence here, the synthesis can start from the top point to the down. Now here, similar to the Sanger or the sequence method, fluorescently labeled building block can come here. Only matching one shows up here, and then it gives the signal. And then along line those synthesis, the nucleotide come in and shows those signal, and it can continue like this. That's why this method is called as a sequence by synthesis, and this shows only one cluster of the information, but actually this process can be handled in parallel. That's why in this chip there are hundreds of millions of billions of spots can show up. These are the signals every round of the synthesis. So after one round of the synthesis, it can give one color out of 4. Then it can finish up those reading. Now a lot of sequencing data can be collected. After this, the collecting is finished. Now depending on those sequences, it can be aligned and compared to the known database. And by reading those fragments through the reference, then it's possible to define those fragments belonging to originally which gene. Utilizing this high throughput the sequencing technology, it is literally possible to sequence millions or even billions of sequences at the same time. In comparison to the sequencing by synthesis, there is another technique so-called pyrosequencing. In pyrosequencing, instead of 4 building blocks, one reaction you add only 1 building blocks. And this building block is not fluorescently labeled the artificial building blocks. This is natural ATP. Then next question is, how can you read whether this reaction happens or not? So if your template sequence is T, then when you add A, it may react with this T and then incorporate it into this DNA fragment. Once the reaction happens, there is one byproduct is formed, one pyrophosphate is released. And utilizing this pyrophosphate and combined together with the luciferin and luciferase the reaction, you can generate light. Then once you see the light, now you can tell, okay, that sequence was T, because that react with the A together. Now, these 4 reactions repeated, and the next one is A, then only when you add the T, this kind of a sequence can be generated. So using this method, advantage is you can utilize natural ATP or natural TTP, which should be much cheaper than fluorescence labeled one. But this advantage can be each reaction you should add 1 by 1 rather than 4 altogether. So at least each step you need to do 4 times of more work than sequencing by synthesis. Another disadvantage in this case, in case if it has AAAA, then you need to add 4T together. Now you should distinguish that whether this is 4T is added or 5. So there is some resolution issues. Practically using this method, you can discriminate up to 6 homo sequences. If it's longer than that, now the error chance is too high. Now we are getting into DNA amplifying technique, so-called PCR. I'm sure you have heard many times of this PCR technique. PCR is a polymerase chain reaction. Using this technique, it's possible to amplify very small amount of DNA is a huge amount of amplified the amount of DNAs. This single technique, we can say, it revolutionized those genetic engineering. And this technique, of course, it awarded the Nobel Prize in Chemistry, 1993. And let's look at those mechanisms. In PCR techniques, now first of all, you need to have a sample which may have some DNA and extract out that DNA and then put those into a PCR machine. Then you can get many copies. And utilize those informations, you can do many things. Now basically this PCR technique, this PCR machine is basically thermal cycler. And in this reaction, in this tube, you can add target DNA and then primer. We have seen primer before, right? And then nucleotides, the building blocks, and polymerase, the enzymes. Now everything getting together in this reaction, the reaction tube, and then put that into these heating blocks. Now in this mixture of the sample, there is DNA at the sample, and we can define those target DNA sequences. Those definition can be done by designing primer in both sides. And first step of cycle, we denature them by increase the temperature up to 95 degrees. Then two strands of DNA, they segregate into single strand DNA. Now next step is cool down this temperature down to 50 to 65 degrees. Then the DNA, the double strand forming is happening. Now this primer can bind to each starting point of synthesis. That's how I said you can define the starting point of synthesis. Now DNA synthesis can go, not stop here, even can go further until the template sequence is ended. Now go to second cycle, heating up again, and every template is dissociated into single strand. Now more primer coming in and bind to this starting point. And then extension again, going up to temperature 72 degrees. Now new strand synthesis is happening. This is the second cycle. In the next cycle, denature again. Now you can see here, 1, 2 of these sequences are exactly what you want to have, but other sequences still have some hangover extra sequences here. But after third cycle, your target sequence exactly the same size is getting further and further increased. Now if you repeat this the cycle of PCR, now different lengths, the longer length of DNA is increasing slowly, but actually your target DNA copy number is increasing dramatically, exponentially. So 10 cycle, 20 cycle, and up to let's say 30 cycle, only 30 cycle after about several hours, 4 hours. Now starting from one single strand DNA, you could get 1 billion amplified DNAs. It's an amazing technique, isn't it?